Warm welcome to the new session of molecular biology. Here we are going to deal with the topic of DNA replication in prokaryotes and eukaryotes. DNA itself replicates before a cell enters into mitosis. Complete and accurate DNA replication is integral to the maintenance of the genetic integrity of all organisms. Watson and Crick first reasoned that complementary base pairing provides the basis of fidelity in DNA replication. That is, each base in the template strand dictates the complementary base in the new strand. As the two strands separate, the purine and pyrimidine bases on each strand are exposed. The exposed bases then attract their complementary bases. That is, DNA replication of one helix of DNA results in two identical helix. If the original DNA helix is called the parental DNA, the two resulting helix can be called daughter helix. Each of these two daughter helix is a nearly exact copy of the parental. DNA replication. The first step in DNA replication is the separation of the two DNA strands by DNA helicase enzyme. DNA helicase untwists the helix from specific locations called origin of replication. The replication origin forms a Y shape and is called a replication fork. Single stranded binding proteins, that is SSB, work with helicase enzyme to maintain the parental DNA helix unwound. A replication fork consists of four components that form in the following sequence. First one, the DNA helicase unwinds a short segment of the parental duplex DNA. Next, a primase initiates synthesis of an RNA molecule that is essential for priming DNA synthesis. Third, the DNA polymerase initiates a daughter strand synthesis. And last one, SSBs bind to single stranded DNA and prevent premature re-annealing of single stranded DNA to double stranded DNA. Now look at the table. The first column consists of the enzymes and the next column which stands for the functions of these particular proteins. The first enzyme is DNA polymerases. It is for deoxynucleotide polymerization, and second one is helicases. It is for processive unwinding of DNA, and third one is topoisomerases. Its function for relieve torsional strain that results from helicase induced unwinding, and next one is DNA primase enzyme that initiates synthesis of RNA primers, and next one is single stranded binding proteins, and which stands for prevent premature re-annealing of double stranded DNA, and next is DNA ligase enzyme which seals the single strand nick between the nascent chain and Okazaki fragments on lagging strand. In the late 1950s, Arthur Kornberg successfully identified and purified the first DNA polymerase, an enzyme that catalyzes the replication reaction. DNA polymerases catalyze the one by one addition of deoxyribonucleotide units to a DNA chain. Polymerase activity catalyzes chain growth in the 5' to 3' direction, and 3' to 5' exonuclease activity which removes mismatched bases, and 5' to 3' exonuclease activity which degrades double stranded DNA. If single stranded, the template DNA must be bound to a primer strand having a free 3' hydroxyl group. The reaction also requires all four deoxynucleoside, that is 5' triphosphate DATP, DGTP, DTTP and DCTP, as well as magnesium 2+ ion. Elongation of the DNA chain proceeds in the 5' to 3' direction. The catalytic mechanism likely involves two magnesium ions coordinated to the phosphate groups of the incoming nucleotide triphosphate and to 3 asparagine residues, 2 of which are highly conserved in all DNA polymerases. Many DNA polymerases also have a separate nuclease activity that allows them to correct mistakes in DNA by using a different reaction to remove mismatched nucleotides. These properties of DNA polymerases contribute to the remarkably high fidelity of DNA replication, which has an error rate of less than 10 raised to minus 8 per base pair. Semi-conservative replication. DNA replication is semi-conservative. Each DNA strand acts as a template and synthesis 2 new daughter strands, each with 1 new strand and 1 old strand. This is called semi-conservative replication. Watson and Crick proposed the hypothesis of semi-conservative replication after publication of their paper on the structure of DNA in 1953, and the hypothesis was established by experiments carried out by Matthew Michelson and Franklin Stahl in 1957. E. coli cells were grown for many generations in a medium containing only heavy nitrogen N15, so that all the nitrogen in their DNA was N15. This will give a single band shown in pink color after cesium chloride density gradient centrifugation. Then the cells were transferred to a medium containing only light nitrogen, that is N14. DNA isolated after one generation equilibrated at a higher position in the density gradient. It will give an intermediate, it is shown in purple color. Continuation of replication for a second generation yielded 2 hybrid DNAs and 2 light DNAs, it is shown in red color, confirming semi-conservative replication.